My name is Jesse Gray, and I'm a postdoctoral research fellow in the laboratory of Michael Greenberg at Children's Hospital Boston and Harvard Medical School. The brain develops in close concert with the environment in a way that is very, very poorly understood. So our work focuses on how the environment can change brain development by altering gene expression. And this is particularly interesting because we know a number of diseases of the nervous system in which the brain would normally be reaching a particular developmental stage and things start to fall apart right at the time where we know the environment is very crucial. Right now we're undertaking two major studies on the solid system. The first is that we are looking at in an in vitro model for neuronal activity, which is neuronal activation by potassium depolarization. We are looking at how gene expression changes when neurons are activated. And we're doing that through RNA sequencing on the solid platform. We're also looking at proteins, transcription factors, binding to DNA using chromatin IP or ChIP-seq sequencing. By combining those two data sets, we have already been able to uncover aspects of gene expression controlled by activity that we never knew about before. The two most salient factors or reasons why we chose solid were, number one, we had a great relationship with Applied Biosystems over many years, and then number two, the platform has so much growth potential. It already performs very well, the solid platform, but it has so much potential for improvements in accuracy, throughput. We're very excited about that potential. The impact of the solid sequencer on my research is hard to underestimate. All of my research is based entirely on solid sequencing. We've already seen that it's not enough to do a chromatin IP and say that a factor binds to a gene. And it's not enough to say that a gene is induced. In many cases, it's actually very crucial to show where in the gene does that factor, that transcription factor, bind. Does it bind in an activity-dependent manner? And what exactly is the transcript that's induced by activity? We're right in the middle of sort of the guts of the work that we're doing with solid, but we can already see that we're able to pick out splice forms, we're able to pick out antisense transcription. We have a sensitivity for detecting transcripts that we never had using microarrays. And so it's already clear that doing gene expression studies on solid is an enormous step forward compared with previous technologies. Next generation sequencing represents a potential revolution in medicine. It's bringing down the cost of sequencing a human genome. And while it's not yet at the point that we will sequence everyone's genome as a routine part of medical care, that day is not too far off in the future. And the solid platform brings us closer to that goal.